Welcome, students, to the second part of exercise 42 on the reproductive system. In a previous lecture, we went over the various organs of the male and female reproductive systems, went over some of their differences, but although they are quite different in terms of their kind of overall anatomy, functionally, they share some common features. They're homologous structures. And one of the things that they share in common that no other kind of organ in the body does is that they go through the process of meiosis. So the testes and the ovaries, the two primary sex organs, the gonads, they don't just do mitosis, they do that as well, but they are the only organs in the body that go through meiosis. So meiosis is a very kind of unique, special form of dividing the nucleus that occurs only in our gonads. None of the other organs in the body do this. And what meiosis does is it halves the number of chromosomes that we find in our gametes. So a chromosome is essentially just this condensed DNA packed with some kind of packing material. So it's all of our genetic materials, our genes. And most of our body cells are considered diploid organs, or diploid. They have 46 chromosomes, so they have two sets, 23 pairs each, one maternal set, one paternal set, and so we call those homologous chromosomes. They carry the same genes for the same treats. You get a gene from your dad for your eye color, you get a gene from your mom from your eye color, and so on and so forth. But the gametes, because they're going to combine during fertilization, if they did not have their number of chromosomes, if they had the same diploid number that all of the rest of the body cells had, when they combine, you'd end up with an extra double number of chromosomes. And so meiosis essentially goes through two rounds of division and results in a halving of the number of chromosomes. And so the gametes, the eggs and the sperm, they are considered haploid. They only have 23 chromosomes, essentially just one member of each of the homologous pairs. The other kind of terminology that we have to use with meiosis is the word sister chromatids, and the sister chromatids are essentially the identical replicated DNA. So once your DNA replicates during interphase, then you have the kind of identical DNA. So these here are sister chromatids right here. This gene is just like this gene, etc., etc. Because it's a, replication, identical replication. So the sister chromatids are identical to each other. So meiosis is different than mitosis, although they do share some similar features. One of the things is that in both mitosis and meiosis, the DNA replicates only once. However, mitosis only involves one cellular division, whereas meiosis involves two cellular divisions, and that's how we end up with that haploid number as opposed to the diploid number. In mitosis, there is no genetic variation. The daughter cells, of which there are two, are completely identical to the parent cell. In meiosis, we introduce some genetic variation through something called crossover. And this results, since we have two cellular divisions, in 4 daughter cells that are non-identical because of that genetic variation. So mitosis is more about growth, development, repair, whereas meiosis is a feature of the gonads only. So here you can see a nice kind of chart. So you can see here is our little parent cell. It's 2N, so it's got 2 sets of chromosomes. It goes through genetic replication. So the chromosomes replicate. We end up with the sister chromatids. But in mitosis, you'll see that we only split once. We get 2 identical diploid daughter cells, whereas in meiosis, we get 2 cellular divisions and end up with 4 daughter cells. that are genetically distinct not only from the parent cell, but also from each other. So you'll see kind of two rounds in meiosis. We get meiosis 1 and meiosis 2. The kind of order of the phases is still pretty similar. Prophase, metaphase, anaphase, telophase, but we have two cellular divisions in meiosis. So what we have is prophase 1, metaphase 1, anaphase 1, telophase 1, prophase 2, metaphase 2, anaphase 2, telophase 2. So during interphase, as in mitosis, that is when the DNA is replicated. So our cell that is diploid, 2N, is kind of briefly 4N with 96 chromosomes. Early here in prophase, the homologous chromosomes kind of joined together as tetrads, and this is where we get crossover taking place. So this chromosome might switch some DNA with this chromosome, which might then switch some DNA with this chromosome, and that happens a lot, and that gives us our genetic variation. This is why the resultant daughter cells are distinct from the parent and from each other. During metaphase 1, they line up on the plate. During anaphase 1, the tetrads are pulled apart. And then in telophase 1, we get cytokinesis, we split, and we end up with two daughter cells. Those two daughter cells will each then go through prophase 2. During metaphase 2, they'll line up again, and then during anaphase 2, the actual sister chromatids will separate, and then we'll get another round of telophase and cytokinesis, and we get 4 haploid daughter cells that are distinct from the parent cell and from each other. A nice little video to show you guys, because I don't know about you all, but diagrams are great, but I like to see stuff moving.
Humans produce gametes, eggs and sperm, through the process of meiosis. Here, we'll follow the production of male gametes by focusing on this cell as it goes through meiosis. Let's begin in the nucleus, where genetic information is stored in chromosomes. Most of a person's cells are diploid, with 2 sets of chromosomes. One set is from their mother, shown here in red, and the other set is from their father, shown in blue. Each maternal chromosome has a corresponding paternal chromosome. These matched pairs are called homologous chromosomes. During interphase, chromosomes are duplicated. Each chromosome now consists of 2 identical copies called sister chromatids. Zooming in, we see that each sister chromatid is made up of DNA wound around histone proteins. Each strand coils up into a tight helical fiber. As meiosis begins, a spindle forms and duplicated centrosomes start to migrate toward opposite poles of the cell. Back in the nucleus, the chromosomes are condensing. In meiosis, homologous chromosomes stick together in pairs. The close association of homologous chromosomes allows segments of non-sister chromatids to trade places. This recombination of maternal and paternal genetic material is a key feature of meiosis. After the spindle forms and the nuclear envelope breaks down, microtubules from opposite poles attach to each chromosome of the homologous pair, resulting in a tug-of-war. At metaphase 1, the chromosome pairs are positioned in the middle of the cell. The next stage begins when homologous chromosomes separate from each other and move toward opposite poles. Each chromosome still consists of 2 sister chromatids. This cell began meiosis with 46 chromosomes, but each daughter cell now has only 23 chromosomes. In meiosis II, microtubules from opposite poles attach to the chromosomes, which then move to the center of the cell. Next, the sister chromatids separate, becoming full-fledged chromosomes that move to opposite poles. Nuclear envelopes reform, and each daughter cell divides into 2 cells. We started with a single diploid cell, and now that meiosis is complete, we have 4 haploid cells, cells with a single set of chromosomes. These haploid cells mature into gametes that can contribute to the next generation.
So that is meiosis in a nutshell. And it is meiosis that results in the production of our gametes. So we're talking about 2 separate processes, what we call spermatogenesis, which is the production of the sperm, and oogenesis, which is the production of the ova. We'll look at spermatogenesis first. So the other term for sperm is spermatozoa. It's kind of each individual, kind of what we call functional little sperm cell. There's really 3 steps to spermatogenesis. We start with these kind of stem cells that we call spermatogonium. Each spermatogonium will go through mitosis and develop 2 daughter cells. One of those daughter cells kind of remains as a kind of a precursor cell, so it can kind of continue to make sperm, whereas the other cell will then enter meiosis. Spermatogonia then becomes a primary spermatocyte. After it goes through meiosis 1, it is then considered a secondary spermatocyte, and then once the secondary spermatocytes go through meiosis 2, we get what we call spermatids. Very early on, the spermatids are still relatively unsperm-like looking. As they kind of grow and mature, they'll develop the flagellum for propelling the sperm through the vagina and the uterus. They'll develop their little penetrating heads so that they can kind of fertilize the ovum. These little spermatids then go through something called spermiogenesis, where they actually become then fully functional sperm with all of the various parts that are required for that sperm cell to penetrate the ovum and fertilize the egg. So we started off with one cell here, but we ended up with 4 resultant sperm just from one initial cell. One of the things that I find really interesting about the production of sperm is something called the blood-testis barrier. So between the cells of the seminiferous tubules, there are these really tight junctions. And the tight junctions prevent the sperm from, or at least parts of the sperm, of these developing sperm, from entering the blood, because if they did, they would actually trigger your body's own immune response. Because sperm do not form until puberty, they are completely absent during the development of a male's immune system. And so this results in the sperm not being recognized as kind of a self cell, not being recognized as part of that particular body. And so without these little tight junctions here between the tubules of the seminiferous, the cells of the seminiferous tubules, if the developing sperm ever escaped into the blood, they would actually trigger an immune response, which is kind of interesting. Okay, so that's spermatogenesis. Oogenesis, slightly more complicated. Surprise, surprise, right? Oogenesis is actually a lifelong process, takes a really long time to complete, years and years, and it actually begins in the fetal period. During the fetal development, the oogonia, the little kind of stem cell eggs, they multiply by mitosis within these primordial follicles. So here's our little primordial follicle, and here's our little oocyte. Within the primordial follicles, the oogonia become primary oocytes, and then the primary oocytes actually start meiosis, but get stuck in prophase 1. And so when a girl is born, the woman already has as many primary oocytes and as many little follicles that she will ever have in her lifetime. So she can't make more. Oogenesis stalls and stays there throughout childhood. Some of these primordial follicles will develop into primary follicles during childhood, but then nothing really happens with the oocyte itself. It's just a little follicular development. After puberty hits in a female, then oogenesis actually resumes. So every month, the primary follicles will become a secondary follicle, and one secondary follicle will become a vesicular follicle. It's estrogen and follicle-stimulating hormone that do this, so hence follicle-stimulating hormone, right? It's stimulating the follicles to develop. Once we are in that vesicular follicle, the release of luteinizing hormone kind of pushes the primary oocyte to finish going through meiosis 1, and we get two results: a secondary oocyte and what we call a polar body. So you'll see the secondary oocyte is much bigger than this polar body, and it's the first polar body because we're going to get a couple more. The secondary oocyte starts meiosis 2 but gets stuck in meiosis 2 at metaphase 2, and the secondary oocyte will only complete meiosis if it is fertilized by a sperm. If it does not get fertilized, then the secondary oocyte never completes meiosis. If the oocyte is fertilized by a sperm, it will complete meiosis 2 and will get a second polar body, as well as now an ovum, a fully developed egg at this point. The first polar body will sometimes, although not always, also go through a little splitting. All of the polar bodies will actually degenerate. And so, unlike what we saw with the sperm, where we start off with one cell and got 4 cells as the result of it fully functional, once a primary oocyte goes through all of meiosis, we only have one functional egg. So if we look at the follicle itself will rupture. That is what releases the secondary oocyte to be caught by the little fimbriae of the uterine tubes. The follicle itself becomes what we call a corpus luteum, which is this structure here that secretes progesterone, helps maintain the endometrium, helps actually sustain the pregnancy. If the fertilization does not occur, then the corpus luteum will degrade to something called the corpus albicans, and will essentially not be functional any further. So you can see here's this corpus luteum from this rupturing of the follicle. So oogenesis and spermatogenesis, even though it's the same kind of process with the two kind of stages of meiosis, the number of gametes that result that are functional is very, very different. In oogenesis, we only get one viable ovum, one viable egg with the 3 polar bodies, whereas in spermatogenesis, we get 4 viable sperm. Part of the reason there's such a disparity there, and the divisions within oogenesis are so kind of unequal, is that the oocyte has to be caught by the fimbria of the uterine tubes and then make it all the way to the uterus, and that's about a 6-7 day journey, and so it just needs to be bigger. The ovum itself needs to be bigger, so it has more nutrients and can just last longer than the sperm. So that's the reason we have a little bit of a difference there. The last 2 things I want to talk about really are about the ovarian cycle and the menstrual cycle. Female reproductive anatomy is, or female reproductive physiology is a bit more complicated than male reproductive physiology. Yes, the male reproductive system is, of course, completely hormonally driven. Testosterone is necessary to make sperm, but in the ovaries and the uterus, it's just slightly more complicated. So both of them go through this kind of monthly cycle, typically about 28 days, although not always, and not everybody's is exactly 28 days. It always is a little kind of wobbly one way or the other. So the ovarian cycle refers to the kind of all of the stuff that happens with the maturation of the egg and the follicles. And then there's the uterine cycle, which is what happens to the thickness of that uterine wall in anticipation, essentially, of having a fertilized ovum implant in it. So we'll talk about the ovarian cycle first. There are two kind of phases, with ovulation kind of splitting them. So ovulation occurs kind of mid-cycle between the two parts. So the follicular phase is all that period of the growth of the vesicular follicle. Again, it's only one primary follicle that will become a secondary follicle that will become a vesicular follicle each month. This is driven by increases in follicle stimulating hormone, hence that's why it's called that, right? Stimulating follicles. During this time, that primary oocyte becomes the secondary oocyte. So it finishes meiosis 1. During the luteal phase of the ovarian cycle, this is after ovulation has occurred, so the vesicular antral follicle has ruptured, released the egg. The corpus luteum now exists from that ruptured follicle, and the corpus luteum produces a whole bunch of hormones like progesterone. It also produces some nutrients and stuff like that that would actually sustain the first 3 months of a woman's pregnancy. After that, the placenta will take over. And so it's not needed anymore. But if fertilization doesn't occur, then the corpus luteum will degrade. And as it degrades, it kind of sends a signal that says, okay, hey, we're reaching the end of the ovarian cycle. It's time to start it all over again. The uterine cycle is broken into 3 phases. There's the kind of pre and post ovulation, but then there's also the menstrual phase. So the first 5 days of a woman's uterine cycle is the menstrual phase. Really low levels of the ovarian hormones, but slowly the kind of gonadotropic levels, the FSH and the LH will slowly increase. This is when she is shedding that functional layer of the endometrium as the menses. Days 6 through 14 is the pre-ovulatory or proliferative phase. Estrogen levels will increase. That will lead to the development of a new functional layer in the endometrium. The cervical mucus will actually thin out, so it's easier for the sperm to get through. And then ovulation will occur at the end of that proliferative phase on day 14, usually triggered by a surge in luteinizing hormone. Days 15 through 28 are the post-ovulatory phase, where we get this kind of increase in progesterone from the corpus luteum, kind of in preparation for the endometrium is like, oh, hey, we're going to get an embryo that implants in us. Plug of mucus will form in the cervix that will prevent the entry of any more sperm, as well as kind of debris, bacteria, things like that. So all of these cycles, both the ovarian cycle and the uterine cycle, completely hormonally driven. So you can see, if we look at the ovarian cycle, here's ovulation at day 14. Again, here's ovulation at day 14 in the uterine cycle. So we have the follicular phase, which is the development of one dominant antral follicle. It is ovulated into the uterine tube, and the ruptured follicle becomes that corpus luteum. The corpus luteum produces progesterone, so that's how you kind of see the spike in progesterone here. All throughout the follicular phase, you'll see our levels of estrogen and progesterone are fairly low-ish, whereas the kind of gonadotropic hormones will slowly rise. Ovulation itself is triggered by an increase in estrogen and a really big surge in luteinizing hormone, after which the luteinizing hormone and the follicle-stimulating hormone will decrease pretty precipitously. If we look at our kind of uterine cycle, you'll see here's that menstrual phase where we're shedding that functional layer as our menses. This is our kind of pre-ovulatory phase where you can see the wall of the uterus is getting thicker and thicker, particularly that functional layer. And then here in this secretory phase, we get a really rich blood supply to the uterus, lots of secretions and nutrients, and essentially the endometrium is like, oh, hey, we might get an ovary, or we might get an embryo implanted in us, and we've got to be ready for it. If that doesn't happen, then the uterus will start to degrade, the functional layer will then be shed again, and we just repeat and repeat and repeat and repeat. So you'll see the secretory phase of the menstrual cycle corresponds to the luteal phase of the ovarian cycle. The menstrual phase and the proliferative phase, oh, I keep saying when I say phrase, respond to the follicular phase of the ovarian cycle. So all hormonally driven, both in males and females. The females' reproductive anatomy and physiology is just slightly more complicated. They've just got a bigger job to do, because once the sperm are made and they're ejaculated, like, that's it, they're done. They're going to contribute some genetic material, and then that's it. But the ovum is not only contributing genetic material, but also has to sustain the embryo until it can implant in the uterus. So female reproductive physiology, much more complicated than male reproductive physiology.